So good evening, everyone. I'm Dr. Sandrine Marlin. I will present you my colleague that will talk about gene therapy for hearing loss today and tomorrow. So Dr. Rizlan Lalou, she's an ENT specialized in deafness for adults in Pitié-Salpêtrière Hospital in Paris. And she also implicated in fundamental studies on gene therapy. Nathalie Lunden, Professor Nathalie Lunden, she's a pediatrician ENT, specialized in deafness for hearing loss in childhood, and she's implicated in the first gene therapy for hearing loss. Dr. Margot Serrego, she's a clinical geneticist working in the reference center for genetic hearing loss in Necker Hospital in Paris. Nathalie is also in Necker Hospital in Paris, and I am the coordinator of the reference center for genetic hearing loss in Necker Hospital in Paris. So we will start with the presentation of Rizlan Lalou.
Thank you very much, Sandrine. And thank you to the Iran Cranio for this invitation and this webinar. So I will talk about principle of the gene therapy for hearing and for vestibular disorders. So what is gene therapy? Gene therapy consists in correcting a disease using DNA or RNA. And one of the most used strategy for genetic diseases is the gene replacement. So as you can see here in this scheme, the therapeutic gene that is the transgene and a promoter associated to this transgene are impacted in a vector and introduced in the targeted cells using a therapeutic vector. This therapeutic vector is a viral vector. It is the adeno-associated virus, so the AAV, and this virus has the advantage to be non-pathogenic for human, to have a low immunogenicity, and to have several natural serotypes that could be specific to one or to other different cells. So for the inner ear is a very isolated organ, both by an anatomic organ barrier and also a vascular barrier. So the delivery must be local. And in most of preclinical studies, we use injections through the round window membrane to access the inner ear. You can see here in this immunofluorescence picture that after the injection of a GFP, of the gene GFP that expresses a fluorescent protein, we have inner hair cells, outer hair cells, and also vestibular hair cells that are labeled in green. So that shows the efficient targeting of sensorineural hair cells in the inner ear after this injection in the mouse. Next slide. How far have we come? Currently, there are two strategies that are developed in gene replacement therapy for inner ear. The first one aim to treat degenerative hearing loss by regenerating sensorineural cells using the differentiation factor ATO1. One clinical trial had begun in 2012, but stopped without published data. However, other most recent preclinical studies show possible success of this strategy in preclinical stage. The second one is the most developed one and aim to treat genetic disorder by correcting a specific pathogenic variation. And actually, currently there are several clinical trials underway that aim to treat genetic hearing loss. So in preclinical studies, the next slide, please. Thank you. In the past year, several preclinical studies in mice showed promising results of inner ear gene therapy for genetic hearing loss. And you can see here all the disease targeted with gene therapy. And in all these diseases, we have successful results. So we have hearing that is restored or vestibular function that is restored. But most of these studies were performed performing injections at a neonatal stage in mouse, and only few others, studied the effect of an injection at a later stage, so at a mature stage here, around 30 days of life in mouse. Next slide. But we know that the development of the inner ear is not the same in mouse and in human. Indeed, the inner ear is functional only 10 to 12 days after birth in mouse, whereas in human it is functional at the end of the second trimester of gestation. So we can hear in utero in human. It is therefore primordial to study a therapeutic window transposable to human. So after the inner ear maturation in mouse, after the 12th day of life in mouse. That was the case for the preclinical studies for the FNB9 hearing loss due to the OTOF pathogenic variation. Next slide, please. For this preclinical development, the first challenge was to deliver the whole gene. And the OTOF cDNA is a large gene, and it is 5.9 kb, so it is more than the capacity of one AAV. So we had to use a dual strategy vector. We used two therapeutic vectors, two different AAV, delivered in the same time in the cochlea. The first one carry the first part of the cDNA, and the second one the second part. In the cell, the two vectors deliver the plasmid, and the cDNA recombine, and the whole protein is transduced. So, next slide. So the great success of gene therapy was obtained for the DFNB9 hearing loss. The DFNB9 hearing loss is a recessive genetic hearing loss caused by the mutation of the otoferlin gene. And here you can see that we have a mouse model for this hearing loss. In a wild-type mouse, we have the otoferlin gene that is expressed in all the inner hair cells, and the hearing is normal. And we can see here labeled in green. And in the cow mouse, in the mutated mouse, we have no expression of the OTOF protein. So there is a complete, profound hearing loss since birth in this mouse. So the proof of concept was obtained in 2090. We showed that the gene therapy for the OTOF hearing loss can restore both protein expression and hearing. We injected the dual vectors, so both vectors, in at 30 days of life in the cochlea of cow mice. First, we observed that the gene therapy allows a sustained expression of the otoferlin in the inner hair cell, like the wild-type mice. We see it here labeled in green. And we see also that the hearing is recovered. Here, the hearing is measured with the ABR, and we see that it can be recovered like the wild-type mouse, and only one injection can recover hearing for the whole life of the mouse. Next slide. We also observed that the gene therapy restored central hearing perception. Here, this is a test that shows the comportamental response to sound of the mouse. And we can see that in the injected mouse, the comportament is, the behavior is like the wild-type mouse. And she acts like the wild-type mouse with a response to loud sounds. Okay, thank you.
I will continue with the basis of genetics of auditory neuropathies and DFN-benign deafness. So we consider that for early onset hearing loss, which is not a rare thing, because approximately 1 in 1,000 newborn is born with hearing loss. There is approximately 20 to 30% of environmental causes, and the main one is the congenital infection with the CMVs, the cytomegalovirus, and the rest, so approximately 80% of early onset hearing loss, is of genetic origin, of which the main presentation is non-syndromic hearing loss, so isolated hearing loss, with 10 to 15% of syndromic presentation. And within the non-syndromic presentations, we can have pre or post-lingual onset. It's most of the time sensory neural hearing loss, and it can go from mild to profound. We have more frequently sporadic cases, but we also have familial cases, and it all depends on the mode of inheritance of the disease. So here's regarding this plot, you see the 10% of syndromic deafness, and for the whole rest of the plot, you have the different mode of inheritance of isolated deafness. So only 1% of mitochondrial or X-linked isolated deafness, and the vast majority have an autosomal recessive mode of inheritance, which I will go through just after, and 20% have an autosomal dominant isolated deafness with a lot of genes identified for each of this mode of inheritance. So next slide, please. Yes. So we have approximately 20,000 genes that are used to make our whole human body and make it function. And all these genes are written on the DNA molecules. And these DNA molecules are actually really, really long threads of letters that write all the genes and that have to be condensed to take some less place. And the DNA molecules are condensed in the form of chromosomes. In all the nucleus of our cells, there are 46 chromosomes that are organized in pairs, because we have 23 pairs of chromosomes, and for each pair there is one paternal and one maternal chromosome, and they are organized in pairs that look alike 2 by 2, because for each pair the two chromosomes carry the same genes in the same order. So for all of our genes, most of our genes, we have 2 copies, one paternal and one maternal. This is important to understand the different mode of inheritance of genetic disease. Regarding hearing loss, we have 118 genes cloned that are causal for isolated deafness, and of which 5 are X-linked, 44 are autosomal dominant, and the majority, so 69, are of autosomal recessive inheritance. And we have 3 genes, main genes, identified for isolated auditory neuropathy hearing loss. So this is a little cartoon for to better understand the autosomal recessive mode of inheritance. So both parents actually carry two copies of the genes. One is perfectly normal and one carries a variant or a mutation. So with their normal copy of the genes, they hear perfectly well. And each time there is a pregnancy, there are 4 possibilities of transmitting the different alleles, so the different copies of the genes. There are 3 out of 4 possibilities of transmitting at least one good copy, so 3 out of 4 possibilities to have a hearing child. And the fourth possibility is when both parents transmit the affected allele, so the affected copy of the gene, and this child will have a hearing loss presentation. So next slide. Regarding early onset auditory neuropathy, we have two main presentations. So either isolated auditory neuropathy that are mostly genes that affect the inner hair cells. So they are mostly synaptopathies. They affect the synapse between the inner hair cell and the nerve cells. And we have on the other hand, syndromic auditory neuropathies that affect mostly the nerve in itself. So for the first group, we have a pediatric or young adult presentation. So the most frequent auditory neuropathy is of pediatric onset, and it's linked to the gene OTOF, which is the gene that we are going to talk about today. It's an autosomal recessive mode of inheritance, and this protein actually encodes for a protein that helps the presynaptic vesicles to go to the synapse and actually transmit the signal to the nerve. The PEF-JAK gene is of really less frequent gene implicated in auditory neuropathy, but is also of pediatric presentation. Then for young adults, the main gene implicated is JAF3, of autosomal dominant inheritance, and it causes autosomal dominant auditory neuropathy. So it's a progressive presentation that starts at the end of the second decade of life or third decade of life. Now for the syndromic auditory neuropathy. So syndromic means that the child will have an auditory neuropathy, hearing loss problem, and also other symptoms. So there are again pediatric and young adult presentations. So for the first one, the FDXR gene, it causes in children the auditory neuropathy and also optic atrophy, so of progressive presentation. Then the gene ATP1A3 of autosomal dominant inheritance cause Kaposi syndrome. So it's an acronym of all the different presentation, the different symptoms that will be present in the child. Another syndrome is the Brown-Violeta-Weinler syndrome 1 and 2, caused by these two genes. And what is important to know is that for these two presentations, there is actually a treatment with vitamin B2, because it's the deficit of this vitamin that causes the symptoms. So it's really rare, but it's important to know because we can treat it with a vitamin complementation. And then there is the TMM8A gene that is X-linked, so recessive X-linked gene, and that causes the Mortran-Ebjerg syndrome with, again, auditory neuropathy and other neurological symptoms. Then for young adult presentation, the most frequent one is OPA1. It's dominant and it causes optic atrophy and deafness, and sometimes in the DOA plus presentation other symptoms, and also Friedreich ataxia and AIFM1 that are dominant by triplet expansion and recessive X-linked respectively. Next slide, please. So now to go right into our subject, the DFN-B9 hearing loss. So we call our hearing loss presentation DFN for deafness, A or B or X for autosomal dominant, autosomal recessive, or X-linked transmission, and then the number is the order of discovery of the different genes. So DFNB9, it is linked to the OTOF gene, and it has two main clinical presentations, either a bilateral profound congenital deafness with initial conservation of auditory of acoustic autoemissions. So when we have neonatal screening with only the AOE, we can miss these children even if they have profound hearing loss. And the other presentation is early sensorineural progressive hearing loss that can also be fluctuating or thermosensitive regarding of the different mutations that are present in the child. And we have auditory neuropathy because we see a desynchronization of the ABR with preserved acoustic autoemissions. And when we do audiograms, we see that the vocal audiogram is way worse than the tonal one. And this is because these children can hear pure sounds, but they have really big of trouble to understand what they are hearing. So this protein, as I said, it has a role in the presynaptic as a presynaptic vesicle conductor, and it helps with coupling with calcium channels to make the exocytosis of the neurotransmitters into the synapse. We need two variations, so one on each copy, so B allelic variations on this gene, and it is of autosomal recessive transmission, and over 100 variants have been described to this day. And we can have some kind of genotype to phenotype correlation, because when children present with two loss of function mutations, so two very severe mutations with a high impact on the proteins, or no protein produced, they present rather with congenital bilateral profound hearing loss, whereas when they have two missense variants, so we just replace one amino acid by another, so the protein is impacted, but less so. So they have early onset hearing loss, but that can be progressive, fluctuating, and with specific variants, there is a thermosensitivity. So each time the child has a fever, for example, they hear way less, and when the fever is resolutive, they go back to their anterior threshold. So these are the two main presentations for this hearing loss.
Thank you very much for this introduction. I will continue on with the more clinical field in our patients, and talk about the current gene therapy that is running. Sorry. So gene therapy, as Rislen explained, is always already used in human. It's either used in vivo or ex vivo. In vivo, the vector carrying the therapeutic DNA is directly injected into the subject or the organ. Whether in ex vivo therapy, the cells are treated, extracted, treated, and re-injected, which is mainly used in oncology. In children or adults, pathology are currently treated with gene therapy using the AAV, such as beta thalassemia, hemophilia, infantile spinal atrophia, or achondroplasia. The inner ear wasn't the target initially for those gene therapy. We had to develop a mouse model, and we had to develop the roots to be able to inject directly into the organ. But nevertheless, even there were some pitfalls. The inner ear remain an ideal target for gene therapy, as it is an isolate organ, so we expect limited diffusion and limited off-target effect. There were some surgical routes already developed in the routine surgery, such as round window, valve window, canalostomy. We had to improve our knowledge on the diffusion of the therapeutic agent around the cochlea and the vestibule, and we know that we might need to have mixed routes to inject to enhance the diffusion. The volume remain pretty small. We are talking about drops, few drops that has to be injected in a very slow manner. So the first human application were done in spring 2023. All the gene therapy clinical trial are phase 1-2 trial, and they are directed to the same target, which is DFNB9 auditory neuropathy. So it is described, this very specific auditory neuropathy, DFNB9, is expected to be about 5 to 10% of all hearing loss. As Margot explained, we can see various profiles. Hearing loss can be mild to profound in pure tone audiometry. When there's residual hearing, patient will suffer poor speech intensity rate, but they all have this synchronized auditory brainstem response, no matter which hearing threshold in pure tone audiometry we can see. And they usually have preserved otoacoustic emissions. So as Margot said, those pathologies are not identified by OAS. The standard of care is currently hearing aids for children that have, or adults that have mild to moderate and even severe hearing loss. But for severe to profound, it's mostly cochlear implantation. And even for some of the children have moderate hearing loss, sometimes we also need cochlear implantation. The other etiologies can be acquired and central, or the nerve can be involved. I will not go through this again. So the first DFNB9 candidates, why is it the first and for now the only one? This group is a very specific and we had the most robust information that we could move from mouse model to human. As it is an isolated synaptic dysfunction, we know that we have preserved inner ear spine structures, which is mandatory to be able to restore a full natural hearing. The mouse model is relevant, and the therapeutic window is on the mature mice, which we can think of moving to a young baby injection at an early stage in human. So there are 5 clinical trials running worldwide. The phase 1 study the body's tolerance of the drug and try to determine the dose and administration protocol. And the phase 2 will determine the dosage where it's the most effective. There has been already a publication in the Lancet early 2024 from the Chinese group, which in this paper injected 6 patients. 5 patients out of 6 had improvement in the PTA, the ASSR, which is a kind of ABR, and the ABR, the auditory brainstem response, which could be seen mainly from 13 to 26 weeks of follow-up. And those were pretty robust results. In April 2023, specifically in Europe, the Sensorion Group, which is a biotech, French biotech, we opened in Europe the first center for this gene therapy, Audiogen. This is a multicentric clinical trial. There's another center that is partner, which is in Australia, Sydney. And we are on the way to open other centers in Europe. So the inclusions are ongoing. I can't say more right now, but we'll have more information, should be at end of this month and early 2025, but we already had 2 patients injected. The candidates in this study are babies that run from 6 to 31 months that have DFN-benign, biallelic autophalanx mutation. They have severe to profound hearing loss, and they don't have yet any cochlear implant. So this gene therapy will be proposed before as an alternative to cochlear implantation. It is a unilateral injection, and we look at, of course, side effect, tolerance, and efficacy, and there will be short, mid, and long-term survey for those patients. We have developed a dedicated injection tool to ensure safety and reproducible injection throughout all the patient and the centers. So those are really very uplifting results, and we have already, as clinicians, to rethink the way we look at those patients. So what are the current impact for those patients? The impact will be through all the professional that will take care of children, deaf children. So it might be pediatrician, ENT doctors, audiologists, hearing aids, feeding, geneticists. So already for patients that suffer hearing loss due to a tough mutation, as we expect gene therapy that could become an alternative to cochlear implantation, we need already to have strong counseling for parents with children and babies that have a new diagnostic of this pathology. So ENT doctors, audiologists, and geneticists should be aware of this new option, and parents might be interested in already getting involved in those clinical trials. We also have to rethink the way we screen children with hearing loss. We know that otoacoustic emission will not screen those babies in the maternity. And so if so, patient and children will arrive with a delay at diagnosis for this pathology. So we need to be aware and maybe push for changing our habits, tools, and regulation for hearing screening toward ABR. Also, as genetician, we might rethink, the groups might rethink the way they screen, the early screening for hearing loss in general, and maybe include at a very early stage this look for DFN-benign. So in general, patients with DFN-benign and other profile, not only hearing severe to profound, but maybe unilateral patient with already a cochlear implant on one side and no cochlear implant on the other side, or mild to moderate hearing loss with DFN-benign might be on the mid and long term possibly candidate for this gene therapy. So we might be a bit more cautious when counseling parents toward cochlear implantation, maybe discuss the possibility of implanting one ear only and wait for the gene therapy to grow and show more robust results, and maybe alter a little bit our practice currently. And in the future, as Ghislaine has showed, other candidates for gene therapy might be available, such as GJB2, USHR2, stereocylin. So we have to look carefully at what will be going on in the next future. Thank you.
Thank you. So now we have some evidence of the success of this first gene therapy for hearing loss. So we'll hope to develop others in the future. So as Margot says, most of the hearing loss have genetic origin when they are the onset it's at birth or in the first years of life. Like you see in this slide, most of them have autosomal recessive mode of inheritance, but there is also dominant isolated deafness, and of course, some cases with syndromic presentation. Next, please. So when we look at the function of the protein that is implicated in this genetic hearing loss, there is a lot of different functions, but most of them are implicated in hair bundle development or functioning. But as otoferlin, there is also proteins that are implicated in synaptic transmission between hair cells and neurons. There is also some proteins that are implicated in cochlear ion homeostasis. Like one of the most frequent presentation due to SLC26A4. There is also proteins that are implicated in oxidative stress or metabolism or mitochondrial function. There is some proteins that are implicated in transmembrane or secreted protein and extracellular matrix, and few of them, and they are implicated most in syndromic deafness, are transcriptional regulated implicated. So it's next one. So if we look to the two more frequent presentation for early deafness, most of them have an autosomal recessive transmission in the family. They are prelingual, but they can be responsible for different severity and progression of the hearing impairments. They are that can be associated also to cochlear or cochlear vestibular malformation, and the more frequent one are GGB2, SLC26A4, stereosilin, and otoferlin. And about 13 different percentage of early deafness are have a dominant mode of inheritance. They are early postlingual, and most of them have a progressive evolution, and there is no cochlear vestibular malformation, and the three more frequent forms are due to pathogenic variation in WFS1 gene or GGB2 or TECTA gene. Next one, please. So if we want to develop new gene therapy in the future, we have to look at several points. First, we have to look at the target cells that are present or not at the time of the treatment. So is there a problem of development of these cells, or an apoptosis of this cell during the progression of the hearing impairment? We have also to know precisely where is the site of expression of the deficient protein and the cell type. It's very important for gene therapy, in particular, to have the good vector to introduce the new gene. We have to look also to the mutational mechanism. It will not be the same if it's an haploinsufficiency or dominant negative pathogenic variation, or if the hearing impairment is due to the overexpression of the abnormal protein. And we have to know what are our goals. We want to stabilize the hearing impairment, or we want to restore the hearing, and we have also to look what we want to do regarding the outcomes of the prosthesis, of the hearing loss, or the cochlear implantation. And also, we can think that in the future, we will be able also to restore the vestibular function. And one thing that is very important is that we have to look to the population that are affected with hearing loss, and we have to do some studies about their feeling about the gene therapy and other new therapeutics, and regarding also the date of the onset of the hearing impairment and the severity of it. And I think that's the end. So you have our email, but I think there is not the one of Rieslen, but we can give it to you if you want. And we are ready for questions if you want. So I don't know if there is any question in the chat. There is no question in the chat question. And there? No, there is no question. Do you have some question? No?
No, there is no question in the chat, but maybe people would like to ask some questions.
Okay. Well, do you see I think you see our emails that are there, and if you have questions also for Rieslen, we can transfer to her No problem. Sorry, Ghislaine.
No problem. I can write my email in the chat if.
Oh, that's a good idea.
Thank you.
That's a
If
very
you
good
want.
idea. Thank you.
So thank you to the ERN to give us the possibility to present this webinar. So if you, yes, there is a question. Do you expect treatment to be limited to one injection in the future?
So maybe Ghislaine, you want to answer?
We know then that in mice it's sufficient. One injection is efficient for restoring hearing in the whole life of the mouse, and it is really stable. We think that the gene therapy, the transgene, is in the cytoplasm of the cell, and the sensorineural hearing hair cells are not reproducing, so it can be in the cell in the for a long time. But in human, we don't know actually.
I think what has been seen is the equivalent of 40 years of lifetime of humans. So hopefully.
Okay, no more question? If there is not, we will finish our webinar, and thank you to Inga to have, there is the new, a new question, any other gene being tested for therapy? So there is preclinical studies, but there is not another ongoing clinical trial.
Yeah.
I could add that this is a very rare disease. So it's one of the problem of enrolling a number of sufficient patients to show the efficacy. But we hope that already the work we've done with the enhancing the surgery, the surgical approaches, and looking at those babies when they have been injected, how they react to the injection. Already this brings a lot of information for the next therapeutic agent that would be developed. And I think also the first result that show improvement of the hearing, it raises a lot of hope and maybe more energy and money coming in for those research. So that I think that will enhance all the work that has been done previously for other genes, for other pathologies.
So there is a question about the percentage of people concerned at the long terms for treatment by gene therapy. I think that for congenital deafness, DFIN-B1 was the perfect example to start, because the cells are there at birth, and so for congenital deafness, it will depend completely on the presence and on the vitality of the hair cells at birth. But we can expect that for progressive hearing loss, we will have a lot of development for other gene therapy. And the question of Anne, about genetic screening. So, of course, I think that this possibility of gene therapy could affect the possibility to for development of genetic screening at birth, because if we want to treat before the cell apoptosis, it will be very important to detect the genetic hearing loss, but also the genetic cause at birth or in the first month of life. So I think it will modify the genetic screening in the future, and there is several programs in Europe and in the world looking for the efficacy of this program for hearing loss.
Thank you, Sandrine. And yeah, I think if there are no any other questions, I would like to thank you all for organizing, first of all, this webinar and participating in it. And I think we highly appreciate it, and would like to thank all the participants who have been here today with us. And if they have any questions, they can always send it to us and or to you